Hi everyone, I'm Thuy Nguyen and welcome to the online version of my video poster. Today I will be presenting a high-level overview of the manufacturing process and controls of our antisense oligonucleotides. While the ASO manufacturing process for nano-rare patients is not unlike that of mass-market ASO manufacturing, it does have a unique set of challenges. One, typically drug manufacturing takes years for one singular drug, whereas our nano-rare patients require these treatments in a timelier manner while maintaining the consistency and quality of good manufacturing practices. Second, the manufacturing scale for nano-rare patients is significantly smaller than that of mass market scale, and thus equipment would have to be appropriately sized for the process. Our approach begins with careful selection of approved CDMO partners with capabilities for our process. Next, we utilize a platform approach due to the similar nature of our ASOs, which would standardize documentation, manufacturing, and testing, which improves operator efficiency and minimizes errors. The subsequent slides will explain the ASO manufacturing process and several of the tests and controls we place to ensure a high-quality ASO is delivered on time. The antisense oligonucleotide manufacturing process begins with amidites, which are the building blocks of oligonucleotide synthesis. Amidites come in various modifications and quality, which will be used in a controlled manner to synthesize the targeted sequence. These amidites are either manufactured or procured through the CDMOs qualified sub-vendors. Once received, the amidites are further tested to confirm they are within specification and in sufficient quantity for the ASO batch prior to manufacturing start. Next, a base solid support is selected for the specified oligonucleotide chemistry. A coupling solution of the first nucleotide base of the targeted sequence is prepared and added to the chain. The coupling solution is prepared in excess to ensure successful coupling. Once coupling is complete, the excess will be washed away using solvents. Next, the chain is treated to aid in stabilization of the newly formed linkage in a step called oxidation. Then, to prevent any unwanted growth, a capping solution is added to ensure no unreactive site remains. Deprotection uses an acid to remove the protecting group on the nucleotide base, which prepares the active site to form a new bond and continue growing the chain. Deprotection, coupling, oxidation, and capping steps are repeated to add subsequent nucleotide bases in the sequence until the desired oligonucleotide is synthesized. Automated synthesizers are typically used, and their inputted recipe sequences are verified by multiple trained personnel prior to start to ensure the nucleotides are added in the correct order. Following the isolation of the desired oligonucleotide sequence, we proceed with downstream processing. Cleavage and final deprotection removes the amino groups and cleaves the desired oligonucleotide from the solid support. Anion exchange purification uses a chromatography system such as a Sactiva ActiPure, where the oligo solution is loaded onto a column and eluted using various buffers. Purification uses a technique called fractionation, which separates the solution based on different biophysical properties such as size, charge, and solubility. Fractions containing product peak are collected and tested on analytical U or HPLC. The ones that pass criteria are taken for ultrafiltration. Filtration removes salts and smaller impurities while retaining the desired oligonucleotide. Lyophilization freeze-dries the collected liquid solution into a dried solid by removing water while the solution is under vacuum. The final isolated oligonucleotide, now in a powdered form, is tested analytically for tests such as purity, water content, identity, sodium content, and microbiome. Once it passes all specifications, it is then stored as drug substance. The tested and released oligonucleotide drug substance is then transferred to a sterile fill finish facility. The dried drug substance is then reconstituted in a formulated buffer, which would be more ideal for drug product administration. Formulated buffers and any intermediate solution undergo in-process testing to verify specifications for pH, concentration, osmolality, and density are met prior to sterile fill. Sterile filtration is a process step which removes or reduces microbial contaminants from liquids. It typically involves utilizing specific filters with extremely small pore sizes. Samples are then visually inspected to assure no defects are observed. Samples passing visual inspection are pulled for analytical testing in parallel, with clinical labeling to reduce turnaround time of release. The vials passing inspection then move on to clinical labeling and packaging, which aid to appropriately identify the drug product and manufacturing information. Once all specifications are met and documentation are reviewed for product release, the packaged drug products are sent to a third-party logistics storage facility and ultimately to the pharmacy of the institution treating the patient. In parallel, the manufactured batch undergoes routine testing to ensure the product is stable and meets all specifications annually. Here are several key metrics we utilize to ensure our manufacturing platform is successful. First, the number of approved CDMOs have grown from 0 in 2020 to 5 in 2025. Second, we can see in the first chart that we have increased the number of new drug products manufactured year over year. The 2025 year is still ongoing, thus the incomplete number. The second chart shows that while we have increased the number of our new drug products manufactured every year, but we have also brought down the time to release. This means our platform approach has allowed for a more efficient and consistent manufacturing time. We have also reduced manufacturing defects, which further allows for a more timely release. As the number of completed programs grows, so does the quantity of data available to further support our understanding of the robustness of ASOs. Here, the left chart shows the status of our ASOs as they go from manufacturing to the clinic as of August 2025. The chart on the right shows the number of programs and the years of stability data. As we continue to produce new ASOs to treat more patients, we will continue to collect data on drug stability of the platform. We would like to thank all of our CDMOs, partners, and the whole n-Lorem team who are making this work possible. Thank you.